My name is Lisa Kronstad, and I'm an AP Giannini postdoctoral fellow here at Stanford University. And I work in the lab of Catherine Blish. She's an infectious disease doctor and scientist. And the bread and butter of our lab is how human natural killer cells sense incoming viral infection. And we think of these cells as the first responders of the immune system. So our lab actually studies a number of different viruses, including HIV, Zika, and dengue. But I work in the subset of the lab that studies influenza. And one of the many reasons I became particularly interested in flu biology was a few years back while I was still in graduate school, my mom, an avid amateur genealogist, tracked down the location of my great-great-uncle Gustav's grave. And she'd found out that he'd fought in Italy during World War I, survived being gassed, had made it all the way back home, and then had died. And she sent me the picture of my grandpa next to the tombstone, and that was his nephew, and with the Norwegian flags that they had placed. But it was the date on the original tombstone picture that really caught my eye. And it was May 1918. And that's a date that coincides with the very first wave of the 1918 Spanish flu pandemic. And it made me wonder if he had been one of the 20 to 50 million people who had died of this virus. So back in 1918, what was causing this pandemic was mystery. And the isolation of the flu virus was still more than a decade in the future. But while we often speak and think of this virus as part of the past, it continued to circulate in and out of the human populations and actually contributed some of its genes to the most recent 2009 pandemic virus. And this is a virus that we all likely have antibodies to, especially because the vaccine was not available until after the peak of the outbreak. But even during non-pandemic years, vaccine efficacy can be up to 60%, but it's often down to only 20%. And because of that, virologists and immunologists really around the world are trying to understand how does the immune system fight this virus, and then to apply that knowledge to both develop new vaccination or improved vaccination strategies and therapeutic interventions. So there's two major arms of the immune system. One is the innate, and the other is adaptive. And we know from animal models that these both have to work together to provide an effective flu response. And while we have a pretty good handle on how the adaptive immune response works, it uses virus-specific receptors, an understanding of how the innate side, which natural killer cells are the major player, how they're able to recognize the virus without virus-specific receptors, that understanding has really lagged behind. So what we do know is that natural killer cells stochastically express this pretty intimidating array of receptors. And the sheer number of these receptors has really been at the core of why NK cells have been such a hard nut to crack. So as often happens, development of new technology has helped us at least partially bypass this problem. So this technique is called mass cytometry, and it couples classic flow cytometry that many of you may be familiar with mass spec. And this happens at a single cell resolution. Historically, immunologists have been limited to about 12, maybe 18 channels, and that's really insufficient to truly delve into the complexity of NK cell biology. So mass cytometry, which I've spent a lot of time in front of this machine, it's expanded this to 40 channels. So this is amazing, 40 channel resolution. And this has allowed us to ask, for the first time, what is the combinatorial receptor profile of NK cells responding to viral infection? And what do these NK cells actually look like? So we've applied this technique experimentally to natural killer cells responding to both that pandemic H1N1 strain from 2009 and H3N2 from 2011. So there's two evolutionary distinct strains that have recently swept through the human population. So we're currently mining the wealth of data generated from these experiments, and this is in collaboration with the Department of Statistics here at Stanford. And few key discoveries have emerged so far. First, we found that NK cells are capable of rapidly and potently suppressing infection of even these very different strains. So this is good news for trying to fight a virus that's constantly mutating. Second, we found that NK cells are capable of robustly producing interferon gamma. And this is a molecule that's absolutely critical for crosstalk with the adaptive immune system. And finally, we've identified two cooperating proteins that are required for influenza to activate NK cells. And we're currently investigating in the downstream signaling network initiated by these proteins with single-cell RNA-seq and phosphoflow. So So taking this work a step further, we're using reverse genetics to engineer influenza mutants to tease out which component of the virus is necessary for NK cell activation. And our hope is that this work and others like it will lay the groundwork for harnessing the cell type to treat patients infected with influenza or with other viruses that are capable of using rapid evolution to escape adaptive immunity.